All right, let's dive into a bacterium that you've definitely seen on the bench, Corynebacterium accolens. For a long time, most of us probably wrote it off as just another skin commensal, maybe a contaminant. But as we're about to see, its story is so much more complicated. It's a true microbial Jekyll and Hyde. So that brings us to the big question, right? Is Corynebacterium accolens a protector, a helpful neighbor living in our microbiome? Or is it a pathogen, an opportunistic wolf in sheep's clothing? The answer, incredibly, is both. And figuring out when and why it decides to switch teams is one of the key challenges for us in the clinical lab. And this dual identity has a name. It's called the Commensal-Pathogen Paradox. It's a term that perfectly captures this tightrope that the organism walks, balancing between being a totally beneficial part of our normal flora on one hand and causing really severe invasive disease on the other. So what this all boils down to is that context is absolutely everything. When you isolate Corynebacterium accolens from a blood culture in, say, an ICU patient with a prosthetic heart valve, that means something completely different than finding it on a skin swab from a healthy person. And that's where we come in. It's our job to interpret that context. Okay, so let's start with the good side. In its day job, Corynebacterium accolens is actually a model citizen of our microbiome. You find it on our skin and our nostrils, and it's not just passively hanging out, it is actively protecting us. And it's pretty clever how it does it. Check this out. It's a really elegant protective mechanism. Basically, Corynebacterium accolens has this enzyme, LipS1, and it uses it to break down the fats on our skin for its own energy. But a key byproduct of that process is the release of these free fatty acids. And it turns out, those fatty acids create a really hostile environment for Streptococcus pneumonia, a major respiratory pathogen. It's an amazing little bit of biochemical warfare that totally benefits us. And it doesn't stop there. Studies show it also takes on Staphylococcus aureus. It actually inhibits the growth of both MSSA and MRSA and can even mess with its biofilm formation. And get this, it does this with a completely different weapon, a protein-based compound, which just goes to show how versatile this little guy can be as a guardian of our microbiome. But, and this is a big but, that is only half the story. When you give this helpful neighbor an opportunity, it can become a really formidable foe. If our immune defenses are down, or if the bacterium gets into a sterile site where it has no business being, its pathogenic side comes out in a big way. And I'm not talking about minor stuff here. Look at this list. These are some of these severe, deep-seated infections that Corynebacterium accolens has been definitively linked to. We're talking endocarditis on both native and prosthetic valves, ventilator-associated pneumonia in the critically ill, even bone infections like osteomyelitis. This is a pathogen we absolutely have to take seriously. So that brings us to the lab. Given this dual nature, getting a definitive species-level identification of Corynebacterium accolens is completely non-negotiable. So how do we actually do it? How do we solve the mystery and make sure we're not confusing it with one of its less troublesome relatives? Well, the first clues are classic micro. Under the scope, you'll see those quintessential corneaform shapes. You know the look, the club-shaped rods forming those V shapes or Chinese letters. And on the plate, its lipophilic nature is the big tell. It grows pretty poorly on standard media, but it'll show that textbook satellitism, growing nice and strong right next to a streak of Staphylococcus aureus, which is basically feeding it the lipids it needs from the lysed red cells. Biochemically, it's got a pretty distinct profile. Catalase positive, nitrate positive, but it's negative for urease and alkaline phosphatase. That negative alkaline phosphatase is really critical for telling it apart from the very similar Corynebacterium meigenlii. But, and this is a huge heads up, commercial systems like API Coronae are notoriously unreliable here. They have been known to misidentify Corynebacterium accolens as Corynebacterium meigenlii. This means we absolutely, positively cannot stop at the biochemicals. For a definitive ID, we have to go molecular. Look, 16S-rRNA sequencing is our bread and butter for a lot of things, right? But for Corynebacterium, it often just doesn't have the horsepower to separate these really closely related species. And that is precisely why RPOB gene sequencing is the true gold standard. The RPOB gene simply has more sequence variation, which gives us the discriminatory power we need to make an unambiguous call. Of course, let's be real. The true game changer in the day-to-day lab has been MALDI-TOF mass spec. This technology has brought rapid, reliable, species-level ID of these tough organisms into our routine workflow. You get a solid score of 2.0 or higher, you can have high confidence in your ID, and you've turned a multi-day workup into a matter of minutes. It's fantastic.
Okay, so we found it, we've got a solid ID, and it's clearly acting as a pathogen. Now what? How do we treat it? Well, this is where things get really tricky, thanks to some unpredictable resistance patterns and, frankly, a major lack of standardized guidance.
And here is the absolute core of the problem when it comes to susceptibility testing. EUCAST does not have specific clinical breakpoints for Corynebacterium accolens. So what does that actually mean for us at the bench? It means we can determine an MIC, but we can't just slap an S, I, or R on the report. We're forced to rely on interpreting that raw MIC value in the full clinical context. And just take a look at the surveillance data. It really paints a picture of why you have to do isolate-specific testing. For the whole group of non-diphtheric Corynebacteria, you see just abysmal susceptibility rates for common drugs like penicillin and ciprofloxacin. I mean, it's terrible. But in stark contrast, Vancomycin and Linezolid show nearly universal activity. This makes that initial choice of empiric therapy a really high-stakes decision. So based on what we just saw, the recommendation for a severe, life-threatening infection like endocarditis is pretty straightforward. You've got to start empiric therapy with either Vancomycin or Linezolid while you're waiting on those lab results. Then, as soon as those definitive AST results are back, the therapy must be tailored to the most effective, narrowest spectrum agent you can possibly use.
When we look at the official guidelines, the general international consensus really lines up with that empiric Vancomycin or Linezolid approach for those severe cases. In the Netherlands, the SWAB guidelines don't specifically mention Corynebacterium accolens, so in that case, clinicians would have to rely very heavily on the AST results from the lab and then extrapolate from guidelines for similar infections, always, of course, in close consultation with a microbiologist.
Now, for pediatric patients, the puzzle gets even more complicated. Treatment is generally just extrapolated from adult data, with the dosing adjusted for size. But this is where we have to circle right back to its protective role. Remember, Corynebacterium accolens in a child's nose helps keep Streptococcus pneumoniae away. So telling the difference between a harmless colonization and a true infection becomes an even more critical and sometimes difficult judgment call in kids. All right, let's start to wrap this up by taking a quick look at prevention and then summarizing the key takeaways from our deep dive into this really complex organism.
The key thing to remember about infection prevention here is that it's not about big, dramatic isolation procedures. This is not Corynebacterium diphtheria, no droplet precautions are needed. The entire strategy just boils down to preventing the organism from getting from a place where it belongs to a place where it doesn't. That means a relentless focus on the fundamentals, meticulous hand hygiene, and a flawless aseptic technique during any kind of invasive procedure. Just a quick note on sources. This explainer was developed using Gemini Deep Search, and the images are from Microbe Canvas. For a full list of citations from all the source material, please check out the notebook document that's linked below. And hey, if you found this useful, we'd really appreciate it if you'd subscribe and let us know your thoughts down in the comments.
I want to leave you with this quote from the source material because I think it just perfectly captures the essence of this whole story. It says that MALDI-TOF and RPOB sequencing have unmasked this hidden burden of disease. The point is this: Corynebacterium accolens isn't some new pathogen. It was probably causing these infections all along, but it was hidden in plain sight, dismissed as a contaminant because we just didn't have the tools to see it for what it truly was. And that leaves us with a really fascinating question to think about as we go back to our benches. As our diagnostic arsenal, from mass spec to metagenomics, just keeps getting sharper and more powerful, which of the organisms we currently dismiss as common contaminants will be the next one to be unmasked as a significant hidden pathogen? Thanks for joining me.